Today, Megan Porter from the University of Hawaii Manoa will be presenting her research on subterranean arthropod biodiversity, ecology, and evolution in Hawaiian lava tubes. Her research is being done through a permit with the HETF. Please note that permits are required when entering the HETF for research purposes. So Megan, whenever you're ready, you can start.
Hey, thank you, everyone. First, thank you for the invitation to speak and tell you a little bit about the research that I've been doing and a team of people has been doing for about the last 6 years now. We've been finding some exciting things, so I'm excited to tell you about them. I wanted to start off today, first of all, figuring out how to change my slide. There we go. With, I think, themes that everyone here is probably very familiar with, just the idea that, you know, the islands of Hawaii are incredibly unique biologically. We have an amazing number of really species that are found only here. So it's one of the most biologically diverse regions on Earth. But I think we're also well aware that it is also at great threat. So ranked number 1 in terms of the greatest biodiversity losses to extinction. And that is only, we're continuing to lose species at a very high rate. This is due to an entire suite of threats, including things like the interactions between climate change, disease, invasive species. And so I use this as an introduction to what I want to talk about, because all of these things, the amazing diversity that we know about in Hawaii and our ability to really understand and try to protect what we have from these multiple threats, that all comes from what we know about the surface landscape. Where my research comes in is looking below that, looking underground for these same sorts of ideas, characterizing what we have, what's unique, what's rare, understanding these ecosystems and the subterranean ecosystems, particularly in Hawaii, have been recognized by the IUCN as an ecosystem that needs more attention. And it's often overlooked because it is out of sight. And so that's where the research that I'm going to present to you today is focused. And everything I'm going to tell you about in terms of what we've done to study those ecosystems and understand the ecosystems there, the diverse and unique species that are there, and how that subterranean system is intimately linked to what's going on above ground, feeds into what we know about Hawaiian biodiversity and how we can go about protecting that. So to start off with, let me introduce you to Hawaiian lava tubes. They were first recognized really as harboring unique ecosystems in the early 70s. And that was by work done by Frank Howarth from the Bishop Museum, and then one of his colleagues as well, Fred Stone from Hilo. And they were really the ones who first recognized these unique ecosystems and the unique arthropod communities that were found there, and did all of the work to document that across the Hawaiian Islands. So from their work and from the work of this in particular is from Asa Awe and his master's thesis at Hilo, they were able to really catalog species that are found only in these lava tube systems and catalog those across the islands. So this is work from Asa's thesis showing the islands, how many subterranean species, so obligate only found in lava tubes, have been documented on each of the islands relative to the island's age. And then he also did modeling that includes things like geology, flow of the age of the flow, elevation, humidity, soil development, all of those sorts of things to create a map of potential subterranean habitat across the islands. And the thing to notice here is that the island of Hawaii, the youngest island, has the most documented species as well as the most potential subterranean habitat. And in part, that's because it is still an active island. These habitats are still forming, which means there's sort of this constant process of creating new habitat space for arthropods to move into. And then as islands age, there is a hypothesis that you see a decrease in that species diversity. So what do we know from the work of Fred and Frank and their students? So as I just pointed out, the island of Hawaii has the highest cave fauna biodiversity of any of the islands. So this is sort of where we're starting when I sort of stepped in and became interested in these systems. And from their work, there are numbers out there of at least 44 cave adapted species documented from the island of Hawaii. But when you go back and look, we can only find records in the literature for about 20 described species of those 44 that they documented. So that means there's a big gap in our understanding of those species and our ability to protect them. In terms of being able to protect a species, if it doesn't have a name, sort of scientifically, it doesn't exist. So we noticed this gap and are trying to work on filling that in, as well as finding new species. And basically, as I just said, so many cave species we know now from the work that they've done and literature searches that many cave species haven't yet been formally described, even though from their work we know that they exist. And also from their work, the island of Hawaii is the biggest island. It has the most potential subterranean habitat. There's a lot of potential habitat that has yet to be explored in terms of looking for and describing these unique subterranean arthropod communities. And so that's where my work and the work of the entire research team really started. And we formed what we call the High Cave Project. So Hawaiian investigations of cave and volcanospeleological ecosystems. That's a mouthful, so hence the shortened version, High Cave. And at the beginning, we had these very specific goals. We wanted to find and document those species that hadn't been formally described in the scientific literature, and as well as continue to document any new and rare species and their distributions across the island. We want this to feed into increased understanding of the cave fauna and the biodiversity for conservation planning. We want to understand the evolution of these systems. So there, as I'll talk about a little bit later, there are interesting patterns of distribution. So understanding how different lava tubes on different volcanoes are related is important to understanding the communities that we're seeing. And then also starting to think about investigating how these subterranean ecosystems are linked to the surface. Because again, like the conservation efforts, they're very closely tied in terms of nutrients. And so conserving both, right? You have to think about both ecosystems. To conserve the subterranean, you also have to think about what's happening on the surface. So the High Cave team has focused mainly the efforts in understanding Hawaiian lava tube biodiversity on the island of Hawaii. Again, it's the island with the most documented species that live in lava tubes, as well as has the most potential subterranean habitat. So it has the most and also has the largest amount of area that has been not yet been looked at. And it is interesting because it has 5 volcanoes, some still active, some dormant. This map just sort of shows you the boundaries of those volcanoes on the island, as well as lava flows, more recent lava flows. So notice that Kahala and Mauna Kea, so these are the 2 oldest volcanoes on the island, and then Hualalai, Mauna Loa, and Kilauea. So these are the younger of the 5, and show the most recent flows because of that. So if you overlay, the little fuzzy, but if you overlay that subterranean habitat model from ASA onto the volcanoes, again, it lines up very well with the 3 youngest, most recently active or currently active volcanoes. And so this is really help to guide where we want to look at new systems, and in particular with the understanding that most of the work that Fred and Frank have done is on the sort of helo side of Mauna Loa, as well in systems developed from Kilauea. So that leaves a large swath of potential habitat that hasn't really been looked at biologically. Another interesting thing when considering these volcanoes and thinking about the evolution of these species, there's a really large diversity in timeframes. So because these lava tubes are formed in specific flows, and we know the ages of those flows, we can study arthropod ecosystems that are in caves anywhere from a couple of 100 years old on Mauna Loa, all the way through ecosystems formed in lava tubes that are, 10,000 years old or more on Hualalai. So there's this really interesting time constraint for when the habitat was formed that allows us to ask really interesting questions about the evolution of these systems, both in terms of how species are moving around from flow to flow, how that relates to evolution of new species, and thinking about how these communities are functioning, how these ecosystems, all of these species are coming together for a functioning ecosystem, and how that changes with age of the flow. So as I said, most of the work, the foundational work from Frank and Fred has been focused sort of on the helo side of Mauna Loa and Kilauea. So the flows here in yellow and sort of this brown gray. So when the High Cave team started thinking about this, we wanted to focus in on an area with flows that hadn't really been well documented biologically. And so the beginning of our work has really focused on flows on the other side of Mauna Loa, on the south side of Hawaii Island. So what does this work entail? A lot of the early work was, as I said, simply getting underground and looking to see what was there. Is it the same thing that was documented in caves that Fred and Frank worked on? Are there different things? Are there new species? Are the ecosystems composed of the same species or species equivalents, essentially? How does that, looking at this new system, how does that line up with what we know from Fred and Frank's work? These expeditions that were focused on documenting this biodiversity, as I've said, it's an entire research team that has done this work. And that team is composed of researchers with a broad spectrum of expertise, including geologists, microbiologists, conservation specialists, local caving experts. So it's a diverse team that has really made this work possible. And so these are expeditions that involve a lot of logistics, coming together, figuring out where we need to target in terms of our understanding. There's a lot of trip planning in terms of where are there cave systems known? What flows are they in? How do we sample those effectively across an elevational gradient? And then once we go out in the field, there's also a lot of building relationships with local property owners, with the resource managers, and making sure that we have all the proper permissions, and that we build those relationships up with the people who are managing these caves, who own these properties, so that they also understand. We get that information back to those owners so they understand what their cave systems, what is in their cave systems, and why they're unique and special. So once we've done all that, then the finding the caves part begins. Just to give you an idea, we work, across the elevational gradient from almost sea level up as far as we can get access to caves. And so the landscape across that elevation changes dramatically, which can have impacts on the cave ecosystems in ways that we're trying to understand, from flows that don't have well-developed forests on the surface yet, all the way up into really well-developed ohia forests. Sometimes the entrances are tiny, so a bit harder to find. That's me poking out of a hole into a really extensive cave system, actually. And sometimes those entrances are really large. So just to give you a sense of scale, that's a person there. That's a helmet, an orange helmet of someone coming down into this entrance. And as we transition from the surface into the subsurface, I like this picture because it really demonstrates that tight coupling in terms of energy, and where in the subsurface, once you get out of the twilight zone, so when you're in complete darkness, there's no more photosynthesis, where does the energy to support these systems come from? And it comes from roots. It comes from surface plants sending roots deep, looking for water, or at least deep enough to intersect with the lava tubes. And that is what drives the energy base for these ecosystems, right? These roots both act as conduits for water from the surface, as well, most importantly, they bring organic carbon into these dark places where there is no photosynthesis, to serve as that nutrient base for any arthropod ecosystem. So once we get underground, we really target these areas and lava tubes that have a lot of root development as places that are going to be concentrations of organic input into the system, and therefore concentrations of where we find cave-adapted arthropods. So sometimes those root chambers, as in this picture, are really extensive. So there's lots of habitat to look through. And sometimes there's less roots, and so the roots get a little more attention from the team that are conducting the surveys. But once we get underground, what we're really doing is we're trying to document what's there. And that includes collection of representative specimens of what we see. And I'd like to point out that we're really cautious about what we collect. We want representative specimens, but we don't want everything that we see. So we're very careful to document what we collect as we collect it, because we don't want to oversample these systems. And one of the things we're trying to do is understand the abundance of some of these rare species in caves now, to help us gauge that. But so we're very careful, but we want to make sure that we collect representative specimens, because that's important for identifying what's there, identifying what's new, and if it's new, getting that new species described formally. Along with those collections, we take environmental data so we can understand how the habitat changes, both seasonally, with elevation, with the sort of overall climate of where the lava tube is, whether it's on the Hilo side that receives a lot of rain, whether it's on sort of the south side that's much drier. We photo document everything we collect. So we're very lucky that we have a phenomenal photographer on the team. That's Mike Slay behind the camera there. And so we have good images to show everyone. So most of the really good bug pictures I'm going to show you are Mike's images to really show you these really interesting and fascinating creatures that we're finding. And then everything we collect gets sorted in the field as far as we can, so that we know what we have from that system. We know, what has been documented there. We can link that to what's been described before. We can flag things that we think are new for further study. We can collect things for genetic work to help understand the evolution, but also to make sure that we have representatives from each of these systems that then go into the collections at the Bishop Museum. And we've worked closely with the Bishop Museum on their Hawaiian cave arthropod collections, both to help get it organized and find, to get the data out of those collections in terms of distributions of species, find specimens that haven't been described yet, so we can get names on those things, and then to add what we're doing to that knowledge base and make sure that specimens are represented in the collections for future research as well. So with all that being said about how we conduct these biodiversity surveys, let me show you a little bit about the really incredible arthropod biodiversity that is found in lava tubes in Hawaii. So perhaps the most recognizable bug from these systems are cave-adapted plant hoppers. And this is the species Olearious Polyphemus. It is a species that is found in almost every lava tube that you go in on the island of Hawaii. So its presence seems to be almost ubiquitous in these systems. And so there are really interesting questions about the evolution of the species. How has it moved around from flow to flow and from volcano to volcano? And so this is probably the most well-studied of the cave arthropod fauna that we find in lava tubes. We find a whole suite of other cave-adapted arthropods in these lava tubes, including in the top left, there are cave-adapted millipedes, and these haven't yet been formally described. So these are in that set of species that were documented but not yet described. So we're working on that. There are cave-adapted isopods. So these are crustaceans. There are crickets. This is a Cachynomobius species, and moths. This is a Shrinkia species. And these all are interesting because, again, much like that Polyphemus, you will find these species in almost every cave that you go in every flow, almost every volcano. And these are all detritivores, scavengers, omnivores. So they all sort of, we think, fall into the same sort of position in the ecosystem. The crickets and the moths in particular are interesting because these actually leave the cave sometimes and come back. So they are another way that nutrients can be transported into the cave system. And then there are predators. So this is the biggest arthropod you'll find in the lava tubes. This is the wolf spider, Lycosa howarthi. But it's not the only predator. So I'm going to show you a few more in just in the next few slides. But I want to point out, I haven't pointed out yet, that these are all truly subterranean species. They're adapted to live in lava tubes and nowhere else. So that includes things like loss of pigmentation. So many of them are very pale or white. Loss of eyes. So you can see here in this wolf spider, which on the surface are normally very visual hunters with big eyes. So they have very reduced eyes. And this particular species is most closely related to a species on Kauai, which is endangered, called the no-eyed, big-eyed wolf spider. So these all have the hallmarks of species that are adapted to live underground. The interesting thing about these lava tube systems is there are multiple predators, which suggests that it's, you know, a robust ecosystem with lots of things to eat. So in addition to that wolf spider, there is a cave-adapted species that is really rare and unique to Hawaii. We call it the thread-legged bug. It's a cave-adapted assassin bug, a Nasidiolestes species. And from our work, it was only known from 2 caves near Hilo. We've expanded that distribution around to caves on the other side of Mauna Loa. And it is potentially, again, a new species. Someone is looking at that now. Another arthropod that you find in Hawaiian lava tubes that's really unique to Hawaii, you don't see it other places in the world, is this cave treader. It's a Cavaticovelia species. Again, only known from Hilo side, from Fred and Frank's work. We've expanded that distribution, and it's being looked at now to see if it is a new species. And this is a cave-adapted version, essentially, of bugs you would see on the surface that you might call water striders. So the things that skate around on water surface, they are now underground and adapted to sort of wet surfaces in the lava tubes. And we've also found really this work at the very beginning, we found new species, and each sort of trip we go out, we continue to find new species, which I think says a lot about how unexplored these habitats are and how little we understand about what's in these systems. So the work that the High Cave team has done has resulted in the description this year of a new caravid beetle species from lava tubes called Paratachys a, described by Jim Lieber, and that paper just came out recently. And these are really tiny, like less than the size of a grain of rice. We have found some new species of Cedar Scorpion. So this is an arachnid group. Again, these are very, very tiny and very hard to find. So we've only found 2 or 3 of these. None have been described from the caves that we're working in. And so we're trying to figure out how to get enough of these to describe them, to get them formally described. Another really exciting new species we have is a terrestrial amphipod. This is a Spelearchestia species. And the only other cave-adapted terrestrial amphipod that has a name is from caves on Kauai, and is endangered. So we feel that this is a really important find and one that is currently being described by some experts in Ireland. So we hope to have a name on this soon. And then the plant hoppers, this group that I said is probably the best studied of the sort of insect groups in lava tubes. We continue to find new species of plant hoppers in these lava tube systems. So here's one that is not only a new species, it is also in a different genus. So the one I showed you is Olearious. This is Eolania. So it's a different genus of cave-adapted plant hopper. And this one is currently being described as well. So as I mentioned, so we started this project trying to characterize the biodiversity in lava tubes that hadn't yet been looked at in different places on the island, to try to understand some of these patterns. And what we're trying to move to now that we've done some of this work is to understand what these ecosystems, look for patterns across these ecosystems, both in terms of what species are there, how are they related to each other, how are they distributed across the different volcanoes. So each one of these triangles represents one of the 3 volcanoes where we have data either that we've collected or from Fred and Frank's work. H is Hualalai, L is Mauna Loa, K is Kilauea. And so we've started to do the genetic studies of some of these species from the different flows, from the different lava tubes, to look and try to gain better understanding of how they're related to each other and how they are moving around from flow to flow. So questions like, when a new lava tube is formed, is it colonized by members of ecosystems from older flows nearby? And how are those related across the volcanoes? So we started this work with genetic work on Olearious Polyphemus. Again, the most well-studied, so there has been some previous genetic work done on it. We've added some of our own data to this. And what you see here is just these branches are colored by the flow a particular group of individual plant hoppers was taken from. And this is what we call a phylogenetic tree. So two branches that are closer together are more closely related than, say, this light pink one is to this yellow branch. So it gives us a way to look at the evolutionary history of these different populations of Olearious Polyphemus. And some interesting things are coming out. So first of all, notice, as I said, they're colored by flow that they were collected in. So it looks like geography is important. Things that are close together in terms of flows are more closely related to each other, despite being on different volcanoes or on flows from different volcanoes. And from the work of the previous molecular work, there are some really deep divergences, genetic divergences among these populations that suggest they may actually be separate species in their own right. So the Olearious Polyphemus is a species complex with different regions having different species. So the number of cave-adapted Olearious is probably much, much higher than the single species it's been described as. And from previous work from Fred and Frank, in addition to Olearious Polyphemus, so now I've got these little bugs to show you how many cave-adapted plant hoppers, Olearious plant hoppers, were known from each of these volcanoes previously. So in addition to Olearious Polyphemus, Hualalai actually has 2 additional cave-adapted species described from flows in that volcano. From our work, we've added, as I said, we've got a new cave-adapted species of plant hopper in a different genus on Mauna Loa. We have another new plant hopper species from there that is waiting to be described. And then we were just in the field a few weeks ago in flows in Hualalai, and we found another, what we think is another new species there that hasn't yet, hadn't been discovered before. So the rate at which we're discovering new species suggests that there's a lot that we haven't, even just in this one group, the plant hoppers, that's the best studied across all of these systems. We're still finding new species at a really rapid rate. So there's a lot yet to find in these systems in terms of biodiversity. And we're starting to look again at those patterns of distribution. So for example, the amphipod, the Speleorchestia, we have found this species now in lava tubes on Hualalai and Mauna Loa, but nothing from Kilauea. Similarly, the Caveticovelia have been found in caves from Kilauea and Mauna Loa, but we haven't yet found it on Hualalai. So clearly this could be due to sampling effort, that we've just missed it. But there also could be some interesting patterns of how these species, when they became cave adapted and how they're moving from system to system, that makes the southern flows of Mauna Loa this interesting mixing zone for different communities. And then we have species as well that we've only found in sort of even just one system on one volcano. I think the best example of this is the cave earwig, Anaxylabis howarthi. So this is an incredibly rare cave-adapted species. Before we started our work, 2 specimens were known, one caught in 1972, one in 1977. And just recently, we found the 3rd animal ever seen of this species. So it's super rare. And and so far only documented on Kilauea. So understanding why that is, it's going to be a hard thing to study since we don't find it almost ever, but that's the best example of, we do have species that some are just on specific flows on specific volcanoes. So we have these really complex distribution patterns, and we're trying to fill in gaps in that to give us a better understanding of these systems as a whole and how they're connected. The next step really is, as we start to understand the evolution and how all of these systems move around from flow to flow, we're interested in questions of, do these ecosystems move? Like, does the entire set of arthropods move into a new flow? Are some of them better dispersers than others? On the ecological side of that, we want to understand the food webs. So what is the base? As I said, we think it's the tree roots. But then how does that translate into a food web? Who's eating who? How many trophic levels are there? As I mentioned, there are a lot of predators in these systems, suggesting it's a really robust food web. And so we've started to sort of investigate the ecological side of these ecosystems in several ways. First, we wanted to tackle this idea that, and really show that these tree roots, and show how these tree roots are serving as the base of the food web, and continue to add evidence to the hypothesis that these tree roots are coming primarily from the native pioneer tree, the Metrosideros polymorpha, or Ohia. So the idea that these roots support cave ecosystems, that comes from early in Frank's work, where he recognized that tree roots are the main energy source for these systems. And so we've documented that in a number of ways. The first thing more recently, we've been doing our surveys in the caves. So we do quadrats where we put out, a meter squared piece of PVC. And then for a certain amount of time, we have team members count all of the insects they see in that space. This is an example of people doing quadrat counts. And we do that both in areas with roots and areas without roots. And indeed, we can show pretty conclusively that in areas where there are roots, so these RG zones, each one of these numbers is a quadrat. The colors are different cave-adapted species. So when we're near an entrance, we get a lot of surface things or invasives. But as we get into the true dark zone of the cave, we get many, many more cave-adapted things associated with those roots. Where there aren't any roots, we don't generally find any insects. So somewhat intuitive, but we've been starting to gather data on what that looks like and what the abundance of some of these species are in those habitats. So we can show that the species are closely associated, which species are most closely associated with those roots. The other way we've really tried to pin that down is using isotopes. So you can look at the isotopic signature of carbon and nitrogen in the tissues of animals, and then you can link that back to what you eat. So it really is true. You are what you eat, and your carbon and nitrogen isotope signatures reflect that. You're typically enriched in very specific ways, particularly in nitrogen compared to what you eat. So you can track that nitrogen change up a food web and figure out who's eating whom in an ecosystem. So we've done that in a couple of different ways. First, we've begun to look at those roots as the base of the food web. So we can show using isotopes. It's a little bit hard to parse out here, but there are roots down at the bottom in sort of a darker green, or I guess in the middle, and then the plant hoppers in lighter green. So they show that characteristic enrichment where plant hoppers, these data show that plant hoppers are eating roots. So roots are the food base for the ecosystem, and plant hoppers are the things that are really making use of that nutrient in a way that makes it accessible for the rest of the food web. And then we've also wanted to think about those roots and really continue, as I said, test that hypothesis that they're coming from native trees. So there's this really important link between preserving native forests on the surface and what's happening underground in these unique ecosystems and communities. And we've done that using molecular tools. So for a number of the caves that we've studied, we've taken small bits of root from the systems we've sampled, and we've been able to use genetics to indeed show that they are ohia roots, and not something invasive or some other plant from the native ecosystem. So we really want to understand, is there something significantly different from in the ohia roots that's serving these communities? Does it matter if it's ohia or something else, to begin to understand that link more closely? And this is really important. Just again, there are threats to ohia and to native forests. And in thinking about these threats, there are really significant implications for what that means for subterranean systems as well, if they are also dependent on ohia for nutrients. And so understanding that link, I think, is really critical, particularly at this juncture with the forest sort of under threat, to understand how that might impact subterranean ecosystems. And then the final thing, sort of piece of research that I want to tell you about is just, we've sort of tackled the food web in terms of the link to the surface, and we're doing that in multiple ways and are continuing to test that hypothesis and understand what that means for the ecosystem. But then to really work on understanding the food web of all of the arthropods that are dependent then on those roots as a nutrient source. And again, we're using isotopes to do that work. So you are what you eat. And this again, so each color represents the carbon nitrogen signature space for a different arthropod in the ecosystem. And so you can see again down at the base, the roots in green and plant hoppers around that. Then there's a whole sort of level, crickets, columbella, moths, millipedes are sort of in that next trophic level up. And then, as I said, there's a bunch of predators, thread-legged bugs, spiders, and some of these things are doing often space on their own. So we don't totally understand, how they fit into the rest of this food web. Beetles are out on their own. Amphipods, the cave treaders are doing something different. So this is allowing us to sort of pick apart each of these and where they fit into that food web and try to understand better how that is all functioning. We can translate these data into something that is perhaps a little clearer, and that's just to look at where each of them is on a trophic scale. So translating it more into a line with plant hoppers down here at the bottom, the things that are making use of that root nutrients. Everything else is sort of dependent on that conversion, which is why plant hoppers are so critical in these systems. But I think what this points out is that there are at least 5 or 6 different trophic levels in these ecosystems. It's more complex than those early hypotheses of just sort of roots and then grazers on those roots with a few predators. And there are complex relationships in here that we still need to tease apart about how each of these individual groups is intertwined with the rest. So with that, I'll just wrap up just by reminding you, showing you some of the work, so where we started with trying to understand just the basic biodiversity of some systems that had been or understudied, and finding lots of new species there that we're working on describing. We've started to now have enough understanding at that level that we're beginning to look at the evolution of some of these groups across flows and across volcanoes to understand how these systems are being, when new habitats form, how are these ecosystems being formed, and then really how are those ecosystems functioning and how those are linked to what's going on in the surface. So I'm going to revise where I started and say Hawaii is one of the most biologically diverse regions on Earth, and that includes underground in systems that most people don't think about because they're out of sight. In terms of thinking about that, we still don't know how many species live in lava tubes, and there's the rate of new species discovery is high, meaning there's, we've got a lot of work to do to understand that in order to understand how to conserve it. And the lava tube ecosystems are intimately and directly linked to healthy native forests, and so we need to think about that in our conservation efforts. And that's why it takes these systems, we've been working on this for 6 years. It takes a lot of field work and a lot of work to look at those specimens, get them ID'd, and it just requires boots on, or in this case, under the ground, to understand that biodiversity and get it properly described so that we can understand it and preserve it. And in that bottom right-hand corner, those are my boots after a week of field work underground. So it requires a lot of boots because you go through them quickly. So I want to give a big shout out, as I've said throughout this presentation, that this is a team effort. There's an entire research team that is involved in this work. And it's thanks to all of their hard work that we have been able to accomplish what we have. There are, a lot of conservation organizations that have also supported this work in various ways, and many people not pictured who have helped in the field work. So thank you to all of them. And thank you to all of you for coming and listening today during a busy holiday season. And hopefully you learned something new about subterranean systems. And love them a little bit because they are incredibly unique, rare, and fascinating systems that we still have a lot to learn about. So thank you.
Awesome. Thank you so much, Megan. That was amazing. I just think it's so awesome that we started with Peter Boston's and now we're ending the year with your presentation. So it's all coming full circle and learning more about our cave ecosystems, and that's awesome. So everyone feel free to turn on your videos or ask questions. I know there's one in the chat and I can read that to you now. Roger asks, if you discover something new to you, how do you collect representative samples without unknowingly damaging species survival?
That is an excellent question, Roger. It is making sure that we have a team that is well versed in sort of what the I guess typical community is, what the things we expect to see in these systems are. As I've said, there's sort of this set of arthropods that we see in almost every system that we go in, or many of them anyway. So understanding when something looks different from that sort of expected set. Communication in the field. So if someone sees something new, saying something so that, multiple people can get eyes on it and get an opinion about it. And then collecting, again, a representative set. And then if it does turn out to be something new, when we get it back and can look at it under a microscope and compare it to the things that we know where we found it. So if, say, for example, if we need to get males to get a species description and we didn't do that in that initial sort of representative collection, we can go back and try to target just the things we need. So it's not, we try very hard not to like collect everything we see. We get that representative set, take it back because it's in a cave, it's dark, these things are small, get it back to where we can look at it under a microscope, make some better determinations, get some expert opinions, get them out to taxonomic experts if needed. And then if it truly is something new and we need some more representation for, we can go back for that.
Roger, did you want to speak on that?
Yeah, I didn't really understand. I mean, I was, you were talking about the cave earwigs, of which there's only been three species, 3 individuals identified. I mean, I don't suppose they're the last 3 in the world, but how do you know they aren't?
It's it's a good question.
Once you take them out of the cave and examine them under the microscope, presumably don't return them to whence
No.
they came.
Correct. In cases like the earwig, populations have to be larger than 3 individuals really to reproduce and survive and have genetic diversity. So, the fact that we've collected 3 over the last 50 years, one, it says that the main populations for this species are not in the habitats for sampling. It's probably up in the cracks in the rock that are too small for us to get into. So when we do see them, we're catching individuals that have sort of gotten lost and come out of their main habitat area. So we're not sampling like the main part of the population. So those really rare species where we see every 20 years at this rate, a single individual isn't impacting a population, but we can get genetic estimates from that if we build up enough individuals over time. And the other answer to that is that's why we're doing that abundance. There's quadrats and trying to get abundance of some of these things. So we truly can, right now we're keeping our sampling low. To our best guess, that's something that's not going to impact these species. But by doing those abundance counts, that gives us some numbers to make sure that we're truly at a threshold that isn't going to be wiping out or significantly impacting the species that we're collecting.
Awesome.
Okay.
Before we go to the next question in the next person who raised their hand, I'm going to read one in here and then we can go to you. Mary Metcalf says, what are some factors in determining a new species?
It's a good question. The ones that we are able to recognize in the field, they look different, right? So Olearious Polyphemus, all white, doesn't have eyes, it has small wings. So when we run into something like that Eolania that is reduced in pigment and eyes from a surface species, but still has some pigment and eyes, so it's different from Olearious, like that's distinctly different from anything that's been described in these systems. So the things that we're recognizing are the things that are sort of big, obvious morphological differences. And that's also why we try to work closely and identify taxonomic experts, because each group has special characteristics that are used to try to distinguish species, some of which are you need sort of expertise to recognize. And so they're the big things we can recognize, but there's probably many more that experts in that particular group can recognize. So we definitely target those that we see clearly. And then we do genetics to help us identify potentially new species that we've missed, because we're not necessarily experts in all of these groups. So there's a whole range of things that can be used. A lot of the ones I've presented to you are new, either because we've never seen that thing in caves on the island of Hawaii before, or they're clearly very distinct from what's been described there.
Okay, awesome. And then we can go to, is your name Kaina? Your hand is raised.
Yes, thank you. I hope you can hear me well. Thank you, Megan,
Yes,
for your presentation. I have a question about the size of the population. Do you now have an idea of from all the expedition you've been doing and this quadrat study that you've been conducting? So do you have now an idea of the size of the populations of the different organisms that are in the caves?
So the quadrat study is relatively new. So we are just starting to build up enough data to get a sense of that. And I think it even just from the sort of visual surveys that we've done in the past, it can be some caves you go in and you will see, so plant hoppers are probably the most ubiquitous thing. You will see, many plant hoppers in a root chamber. Other systems you'll go in and you'll see maybe 2 on a particular visit. So it's really variable in ways that we're trying to understand, probably related to, so the roots seasonally probably dry out some, from wet to dry season. It depends on where they are climatically, elevation. So there are factors that are controlling some of that. And I think some of it is, again, we are seeing only a portion of the population. I think a lot of it is up in the fractures and cracks of the rock. So we're starting to get it, but also I should say in those quadrats, we're capturing the things that are most closely associated with roots. So the plant hoppers that eat them, detritivores like isopods, crickets, springtails, but we typically aren't getting the predators in the system. So they are not, as they are probably moving from patch to patch. So we know that we're missing things, and we know that we're missing habitat because of the way we're doing that. So it's a first pass at it, and it's going to give us some good numbers to start with, but it's not capturing the entire community either. So we're still working on the best way to do that. I'm open to ideas if anyone has got some suggestions. I know we can do it genetically, but again, that requires more money and also enough individuals to be able to start to estimate that. And so we haven't gotten that far in the genetics for many of the things yet.
Yeah, to me from,
Sorry, I just wanted to announce, I know it's getting close to 1, so if anybody needs to leave, thank you for coming. But yeah, thank you. Sorry, go ahead.
Thank you, everyone.
It was just to say that from a genetic point of view, not knowing the size of the population, it's complicated, because to really get an idea of the size of the population, it will at least we'll need at least 10 individual per population. Yeah. So
it's tough for the things we're not capturing in those quadrats, like the wolf spiders and the predators. Those are also the things that we see. We collect the fewest of, again, because they're not so tightly coupled to those roots. They're probably moving between patches. So we get less individuals of those overall. And so those are the harder ones that they're going to be the harder ones to really get at this population size question, for sure.
And Sam asked, is there a greater density of arthropods near the cave ceiling where the roots come down through the surface, or near the cave floor where roots continue to make their way through the floor?
Excellent question. So the way that we're doing these counts, we do timed counts, and we will do sort of 5 minutes looking at the floor and then 5 minutes looking at the roots and ceiling. And we haven't broken down the data that way yet, but I would say just sort of anecdotally, you do see things up, like we tend to find, say, thread-legged bugs and cave treaders on the ceiling and near the sort of the tops of roots, walking around on the walls. You tend to find plant hopper, particularly the nymphs, actually on the roots. You'll find springtails and isopods on the roots. And then you find things like beetles, springtails, sort of on the ground around the base of the roots, sort of where you get some detritus. So there's definitely, I think, some finer scale niche partitioning going on there. Right now, we just have all those numbers in bulk, but we are definitely seeing differences in where we find things, and recognizing that we're not capturing all of the habitats for the species we know are there for those counts as well.
Okay, and then I'll get to your raised hands in a bit. I just want to make sure we answer everything in the chat first and then go back. So Jason asked, does the presence or abundance of microbial mats have any impact on the population of different species?
Good question. I would say from our current understanding, maybe. So one of the ideas for when I showed that sort of isotope plot, and I said there were some things out doing their own thing that were away from the plot, the sort of carbon nitrogen space of other things, the beetles, the cave treaders, the amphipods. One of the ideas is that perhaps they are making use of microbial mats as a food source as well. We are going to start testing that. We don't have any of that isotopic signature for the microbial mats in the systems yet, but it's one of the ideas of things we'd like to do. And I certainly think it's possible. We just don't have the data for it yet.
Megan, that would be really interesting to know because we did some checking in the New Mexico lava caves and we found various like mites or Climbula on the algal map, but none on the microbial map.
Ah, okay.
And
okay.
I don't know if it's because of some of their actinobacteria secondary metabolism produced and whether that is antithetical to them or what. But I'd love to know what you find with that.
Well, we'll keep you posted, but that's sort of the next idea for these things that are sort of not in the direct trophic interaction from the roots through the plant hoppers up. Like something else is going on and there's things that are out on the side.
Awesome. Roger, did you want to go?
Yes, thank you. I'm not very clear about how the interconnection of the various lava tube formations and so forth. I mean, the animals that you're looking at don't look particularly geographically mobile, and I'm wondering how they get from one place to another. I mean, if the same species presumably somewhat from one cave system to the next, is it?
Yeah.
But I don't really understand.
So that's one of the big questions we're interested in investigating, particularly using molecular tools to track that down. So there are hypotheses from Frank's work that you have these communities in older flows, and then you have younger flows coming over the top of them, and as that cools, they can just sort of migrate up into the new habitat, right? So you sort of get a movement of these things from older into younger flows into lava tubes as they form. But that's not the whole story either, because you see these things have moved sort of from volcano to volcano. And I don't think we have enough of an idea of how they're related genetically across flows of different ages and across volcanoes to really know what that pattern is. I think it's more complex than that, sort of just continually moving into younger flows. They are clearly moving from system to system through the interstitial cracks in the lava. And we only can see and sample them in those cracks that are the lava tubes that are big enough for us to get into, right? So I think a lot of it depends probably on the development of the root systems. So as Ohia comes into the new lava flows and how extensive those roots are, because they're moving through those cracks, the roots are their food source. If there are roots there, they can follow those roots, through those cracks wherever they can fit. So I think it's tied to development of the root system and the forest above. And then we don't know. That's one of our big questions is how are they moving around from system to system? Is it a simple, just like stepwise fashion as new systems are formed, or is there something more complicated going on? And we don't have an answer for that yet. So it's a good question.
Okay. I don't know whether mahalo means thank you or goodbye, but mahalo.
It is thank you. Thank you. Thank you for the good questions.
And then Kaina again.
Yes, I was wondering if the isolation of the predators on your food web might be a link to some groups that you're not catching in the tubes, or
yeah, that is another possibility that we're missing a piece of the food web, either because we're not sampling in a way that we're catching it. The second possibility is that they're too tiny for us to get isotope data from. Like the springtails, I think we had to pool 10 or 15 individuals to get a signature. So that's, tiny things are hard to use this analysis on. So that's another possibility is either of those limitations could explain why we're seeing something sort of disjunct from the main pattern, for sure.
And then Diana, you can go ahead.
So Megan, is there any work being done on microbial symbionts, possible microbial symbionts?
Yes. So that's I haven't talked any about any of that work. That's been it's really the work of Rebecca Chung, who's a collaborator and a faculty member here at UH Manoa. And so her specialty is really looking at endosymbiont evolution. So she's been really digging into the endosymbionts in the planthopper species, because that is really important for mediating how those nutrients are captured from the roots and sort of translated into animal, which then everything else in the system can eat. The planthoppers truly are little bags of sugar that everything else in the system eats. And they can do that. They can make use of the nutrients from the roots because of their endosymbionts. So she's been really doing a lot of work to understand the endosymbiont evolution, both within like Olearious Polyphemus, as well as in, as we find these new species, what does that look like? And what does that tell us about the way nutrients are transferred through the system?
Okay.
Yeah.
That would be interesting.
Yeah. So I'll have to get her back to talk about that.
Awesome. Thank you so much, Megan. And if there are no more questions, I know we're running over time. I will be sharing Megan's email in my follow-up email, as well as the link to our channel. So you can also watch Peter's presentation. But yeah, are there any more questions? Sorry, I spoke too soon. No? Okay, so thank you everybody for attending. Happy holidays, and I hope you have a great rest of your day.
And thank you for
everyone.
the
Thank you.
Mahalo.
seminar.
Mahalo. Thank you, everyone.
Mahalo.